Thank you. Thank you, Andy. Thanks to the organizing committee for inviting me to share with you some new data on immunogenetics in CLL. You might, of course, wonder if there is anything else to dissect 20 years after the publications of these seminal papers which have changed the way we think about this malignancy. So it's an amazing 20 years where we have learned so much. We have understood how the B-cell receptor is central to the pathogenesis of this disease. And what these studies have shown us is that structure underlies function or is linked to function, and this is also linked to clonal outcome. Let's try to put immunogenetics in context, though, and this morning has been very oncogene or oncoepigene driven or biased, and we did try that. So this is what happens if you try to integrate immunogenetics and oncogenetics. We did that a couple of years ago, and you can see here the typical recurrent cytogenetic aberrations and very frequent gene mutations in CLL. And you can see for yourself, I think it's quite evident, that the outcome of patients with mutated immunoglobulin genes, lacking any major cytogenetic or genetic mutation, is far superior compared to that of patients with unmutated immunoglobulin genes on a similar genomic background. More recently, we had the exact same finding in patients with complex karyotype. In other words, you can have patients lacking any cytogenetic complexity, lacking TP53 aberrations, and they will do much, much better if the immunoglobulin genes are mutated. I think I may challenge you, but an obvious question coming out of these results is whether the immunoglobulin, the B-cell receptor immunoglobulin, might indeed be the ultimate driver. I'm a believer in that, and let's share with you some facts and some speculations as to why. A first fact pointing to the centrality of the B-cell receptor is the skewed immunoglobulin gene repertoire, first shown by Guillaume de Guéraud, Nick Chiorazzi, and others in the mid to late 90s. Importantly, what they also showed was a differential somatic hypermutation status, and depending on which variable gene was expressed in the immunoglobulin. In other words, you could see 169 being predominantly utilized in unmutated receptors, as opposed to the 434 gene, which was far more frequently used in mutated, somatically hypermutated receptors. In the case of 321, which, as you'll see later in my talk, appears to be quite interesting and distinctive, it was a mixed situation. Another finding coming out soon after the initial publications was that about 50% of patients expressing 321 receptors would be highly similar. These receptors would be very restricted. It was a finding by Richard Rosenquist and the Uppsala group that was soon confirmed independently by other groups, and it was Nick who coined the term stereotyped. It's a nice Greek word, means to be repeated with limited or no variation. Now, going back to basic immunology, the chances that you will find two independent B-cell clones carrying the same immunoglobulin and are in this order, meaning, in the end, that chronic lymphocytic leukemia is clearly not an unfortunate stroke of serendipity, and definitely alluding to antigen selection in CLL ontogeny. So stereotypes are clearly the utmost in repertoire restriction in CLL. Do we know all about them? The answer is no. There are many questions. What about their frequency? What is their relation to the somatic hypermutation status? Are there distinctive features of stereotypes that could give hints into distinct pathomechanisms for some disease variants. And finally, what is the clinical significance of stereotypy? I will start with the frequency. So it is now almost 80 years ago when we published that the stereotyped fraction of CLL accounts to almost 1/3 of the entire cohort. This was a series of 7,500 patients, 33% ended stereotyped, and another interesting finding in this study was that you could have many different stereotyped subsets, each defined by a unique configuration. Today, with 31,000 patients, more than 4 times as many as in the previous study, the incidence of stereotypy is an amazing 41%. So the increase in patient number led also to a quite important increase in stereotypy, in the incidence of stereotypy. Importantly, few stereotyped subsets account for a sizable fraction of all CLL. You should think that the possible configurations are trillions, and here only 4 variants, only 4 stereotyped subsets, 7% of all cases. This is indeed amazing. Now, what is the relation to the somatic hypermutation status? It's clear, it was clear, that's published many times since our original publications, that stereotypy is more frequent in unmutated CLL cases. But with the new data in this new, much larger series, the incidence of stereotypy within the mutated, IG mutated category has increased considerably. And there is an obvious, I would say a reasonable speculation. With a relevant increase in numbers, perhaps all CLL may turn out stereotyped. What could happen if we have 100,000 patients or 200,000 patients? Could it be that they will all turn stereotyped? We'll see. However, there is another obvious question. Did we perhaps previously miss stereotypes in the mutated CLL category? And the answer is yes. And why is yes? There are some mutated CLL subsets showing very degenerate CDR3 motifs. This is one case, one indicative case, and you can see here the sequence logo of the CDR3 of many cases in this subset. Now, the height of each letter is proportional to how frequent we observe that particular amino acid within that position in the CDR3. So the R and the D are almost ubiquitous, the G very frequent, but in other positions you see degeneracy. So can I convince you what is the evidence that these are indeed stereotype? And it's not only the example I showed you before, here is another example. And there are many more within the mutated category. So is there additional proof that these are truly stereotype, or is it just that I'm claiming? And the answer is that there is indeed compelling proof for stereotypy, and that they are truly connected. You can see for yourselves, here is the alignment of the entire variable domain. And highlighted in yellow are somatic hypermutations, so changes from the germline. In my view, it's indeed amazing that all these independent clones acquired the exact same somatic hypermutation, the exact same amino acid replacement. When it comes to frequencies, you heard me speak about the 1 in a trillion chance. Here we are talking about something that is almost impossible to happen, and yet it happens. If you need additional evidence, I will highlight to you what is the most frequent mutated stereotyped subset. This is stereotyped subset 4, which is a prototype for indolent disease. And here is the web logo, the sequence logo, for more than 250 cases in this subset. There are particular positions within the CDR3, particularly at the tip of the CDR3 loop at the center, where we see extensive intra-subset heterogeneity. So different clones may carry different amino acids. These are web logos, sequence logos, that I hope you agree with me, look very similar to the one on the left. But these are sequence logos of intraclonal variation. So this is intraclonal data because these are NGS results of many millions of sequences obtained in individual patients of this subset, which makes me share with you this idea that the intra-subset heterogeneity is also reflected intraclonally. Another thing that we notice in this large cohort is that stereotyped subsets can appear very similar. This is an example from unmutated CLL. All these subsets utilize a 169 gene. They carry a very similar CDR3 amino acid sequence, differing simply by one or more amino acids in length. But this phenomenon can also happen in mutated CLL, leading us to introduce a new concept. We call it satellite subsets. So satellite subsets are variants of major subsets, if you will. And I will, in the remainder of my talk, I will give you a quite instructive, I will present to you a quite instructive case of subsets 2 and 169. Subset number 2 is quite interesting for a number of reasons, being the most frequent in CLL, almost 2.5 to 3% of the entire cohort, and the prototype for aggressiveness. So it's very frequent, whereas this 169 is very infrequent. They share very major similarities within the CDR3. They both carry a CDR3 of 9 amino acids in the heavy chain with a ubiquitous acidic at position 3, which is why we call it the 9 acidic 3 CDR3. Is it all that they have in common? No. The V gene is different. In the case of subset 2 is the 321. In the case of subset 169 is 348. But 321 and 348 are 97% identical at the germline level. Nature doesn't care about gene names. Nature cares about structures. Light chains, they are identical, and they both utilize a gene called IGLV321. And another thing that we noticed in subset number 2, this is studies that we did with Paolo Ghia and Massimo Degano and Hassan Juma, is we studied whether the subsets may indeed have a distinctive mode of self-association in the B-cell receptor immunoglobulins. And in the case of subset 2, we found that these immunoglobulins do indeed homodimerize. And very importantly, these homotypic interactions are mediated by IGLV321. In the case of subsets 2 and 169, the somatic hypermutation status is what we call borderline. So both subsets contain both mutated and unmutated CLL. And what about recurrent mutations? There is an amazing incidence of deletions, which is very peculiar, in the CDR2 of subset number 2. Subset 169, we didn't know anything about it until very recently, when Katerina Yemenezi in our group performed an NGS study, and she's presenting it tomorrow during the poster session, showing that all 169 cases analyzed carry the mutation at subclonal level. Another distinctive feature of subset number 2, in fact, the critical change for the acquisition of self-association capacity in the B-cell receptor is a mutation in a strange position, the VL to CL linker. We didn't know anything about 169, and Katerina once again found that it is present in all cases at clonal level. Finally, I shouldn't forget mentioning that subset 2 and subset 169 share an amazing 45% incidence of SF3B1 mutations, meaning that there is a distinctive genomic background for a distinctive immunogenetic profile. And only yesterday, Massimo Degano from Milan, our crystallographer collaborator, shared with me his very, very recent result. The crystal for subset number 169 is now produced. The homotypic interactions are highly similar to those that we observed in subset number 2, and again, the light chain IGLV 321 is the critical mediator of self-association capacity. So I hope that you would allow me this reasonable hypothesis. Satellite subsets could share similar pathophysiology. And could that be clinically relevant? Should we care about subsets clinically? Over the years, we and others have tried to do that. Panagiotis Baliakas showed that you can discriminate patients based on subset membership. That subset 2 is as bad as TP53 aberrant CLL. Then we showed that you can have within mutated CLL different outcomes. Davide and Gianluca showed subset 8 as a prototype of aggressive disease. But in this meeting, and I find this extremely interesting, Sonia Jaramillo, Stefan Stilgenbauer, and the German friends show that subset 2 is an independent marker for unfavorable prognosis regardless of the somatic hypermutation status. And they conclude that subset 2 membership should be proposed for risk stratification of patients, and I propose, interpreting their data, that subset 2 patients do not benefit from chemoimmunotherapy. So there are similar courses and outcomes in these two subsets. These are data that Panagiotis kindly provided me recently. So in the end, I think that there is a plausible argument that satellite subsets could share similar pathophysiology that is reflected in a similar clonal behavior with obvious implications for risk stratification. So if it's subset 2 that our German friends propose for risk stratification, why not its satellites as well? So how to conclude? B-cell receptor stereotypy is clearly the strongest molecular evidence for antigen selection in CLL ontogeny. It is a powerful means for breaking down subsets, CLL into subsets with homogeneous profiles. It has definitely contributed to the identification of distinct pathomechanisms in different subsets. It may prove a key to overcoming the heterogeneity of CLL, and in our view, it is clearly relevant for implementing tailored therapeutic approaches. In closing, I have to thank many close friends and collaborators from both sides of the Atlantic, and also thank you very much for your attention.
Okay, the paper is now open for discussion. Are there any questions for Kostas? Okay, perhaps while you're thinking, perhaps you didn't mention it, but have you looked at B-cell receptor signaling per se in your different stereotypes? And how does the strength of signal differ between the different stereotypes?
Yes, in the interest of time, I didn't present such data because the talk was immunogenetics. But the answer is yes, different subsets signal differently. Here the instructive case is subset number 8, the Richter subset, if you will, where these receptors signal far more intensely, even when we compare them to other aggressive subsets or any unmutated CLL. And we also see distinct patterns of signaling, not only through the B-cell receptor, but interestingly also through toll-like receptors. So you can have distinct TLR signaling capacity and outcomes after TLR triggering in subset 1, unmutated, aggressive, versus subset 4, or other mutated CLL.
Brilliant.
Kostas, thank you very much. So, in terms of the stratification of patients in trials or the reporting of patients in trials, should we be taking subset 2 out of the mutated group of patients and reporting them separately, or including them with the unmutated? How would you recommend we use that?
I recommend that subset 2 is considered independently. And I think, thanks so much, Pete, for giving me the opportunity to say that it's not a simple binary mutated, unmutated. So within the mutated, we have to delve deeper, and subset number 2 is the first clear example of a variant of CLL that has to be considered independently, also in the context of clinical trials. In my view, the recent data of Sonia and Stefan and the German CLL study group is very important because it emphasizes what we previously reported on retrospective material, that the outcome of these patients is very distinctive, and this is clearly independent of the somatic hypermutation status.
Okay, we're actually out of time, but because you stood up so early on, can you just be a very quick question, please?
I try. Do your data support the hypothesis that specific antigens are very important in patients, the sharing the same antigens? And should we look for them?
There is evidence that different subsets have a different antigen reactivity profile. I mean, this is work pioneered by Nick Curazzi, Anders Rosen, and others. So we have shown that, for instance, if you compare the bad ones, 2 and 8, they have a very different pattern of antigen reactivity, but it's not as black and white. There are some examples within the mutated CLL, the broad mutated CLL category, where you can have a very specific antigen reactivity, for instance, a minor subset recognizing fungi, but it's not so black and white again.
Okay, we'd like to thank Kostas. Thank you for a great talk. Thank you again.